Hello, everybody, and welcome back. Today we're going to talk about the forces of evolution. Here we have a lion taking down a Cape buffalo, an example of selection. I don't know if the buffalo is necessarily a healthy one or not. Predators often target the weak, the sick, the young, and the old. However, survival of the fittest is often a term we use to describe this sort of thing. Um, is evolution and survival of the fittest necessarily the same thing? Well, evolution at its core is the changes in allele frequencies in a population over time, generational time. Whereas survival of the fittest is essentially synonymous with natural selection. And natural selection is just one way that allele frequencies and genetic variation in a population can change. So evolution encompasses more than just natural selection. And in fact, it encompasses other forces. So we're going to talk about these today. Mutation, this is where the DNA code itself can be changed by some sort of environmental or some sort of intrinsic factor. And it's the only way to produce brand new genetic variation out of scratch, right? Gene flow, this is the movement of individuals, immigration, emigrations from one population to another, and as they move, they carry with them their genes, thus enriching or depleting populations. Non-random mating, in other words, sexual selection, where certain traits bestow advantages in a mating context, and these tend to become more common over time. Genetic drift, which is when a large population, through some sort of random event, becomes a smaller population. And as that happens, you basically have a random sampling where only a small subset of the genetic variation from that bigger population persists through time. And then finally, of course, natural selection, like here with this bird eating the bluebirds because they're very conspicuous up against the green leaves as opposed to the well camouflaged green caterpillar, right? So those blues are being selected against. So let's start first with mutation. But before we get to that, let's talk about what's going on. Is evolution acting on the individual or on the population? Well, we know that natural selection is working at the individual level, right? Individuals are removed out of a population through selection. And as a result of that, it's operating at the genotype, right? But really what it's doing is it's acting on the phenotype of that creature, right? So if you have an individual with particular traits and they happen to not confer an advantage or they do confer an advantage, you're going to see that impact. But as those phenotypes are coded in the DNA, that's going to be removed. As far as evolution goes, however, we talk about evolution, we're usually talking about changes that are occurring transgenerationally, right? So from one generation to the next, and you're seeing those changes over time. So selection's acting on the individual, evolution's acting on a population. So why is it this way? Well, because a population is where you have genetic variation, right? You might be an individual, but you don't necessarily represent the wide range of genetic possibilities. A population, however, does fulfill this first criteria of evolution, which is the presence of variability between individuals. So you need genetic variation in the population because it serves as a substrate by which evolution is going to function and by which these forces of evolution are going to act. So we'll talk about the first, mutation. Here is a DNA sequence, and you see here the thymine being replaced with an adenine, as indicated by the arrow. Different nucleotides being substituted, and you can tell that it's in response to the radiation coming from UV light. Right? So this is an example of what's called a point mutation. It's something that can have a very drastic impact, have a very positive impact, or more likely have no impact at all. It's just one of many different ways to create new genetic variation from scratch. Again, mutations and the presence of mutation is where genetic variation ultimately starts from, right? The rest of those forces of evolution we talked about are about changing what's already there. So these changes in DNA can produce new alleles, and these are what we call mutations. It's a random process, meaning that there's no way to necessarily direct which areas of the DNA are going to be impacted, and it's impacted by all sorts of different things. And if the conditions are right, you can get a mutation. Are all mutations necessarily bad? The answer is no. We tend to think of, mutants as being this negative thing and turning into some kind of bizarre aberration. Usually it's kind of ridiculous because you'd have to change a lot to really change something really, really prominently like that. But mutations tend to be, if anything at all, they tend to be, good or bad, or more likely have no impact whatsoever, at least anything visible, right? It really depends on where that mutation is happening and what the consequences of it are. Remember that DNA is basically just the code for producing proteins. So as that mutation impacts that process, the more it does impact the process at the end and the product at the end, then it's going to have a bigger impact on the phenotype. So mutations can be really small, such as a single nucleotide being substituted, right, or changed, a point mutation, kind of like I showed you earlier with the thymine being replaced by the adenine. They could be an example of a bigger thing where entire sequences might be added in or deleted, right? Or even just a single point being added in and deleted, right? And when this happens, you can potentially change the length of something, right? You could potentially change the length of a gene. You could potentially inactivate it. You could potentially substitute the amino acid out for something else. So it could also lead to the production of a big effect if enough sequences are thrown out of alignment, produces what's called a frame shift. Or it could be something that has no impact whatsoever. So here, for example, is hemoglobin. This is the DNA molecule for hemoglobin. It's showing the DNA coding for a particular thing that gets translated to produce the normal hemoglobin molecule, which is obviously it's the protein that carries oxygen in the blood. And you see here it's a glutamine's being inserted as the amino acid product of this wild type hemoglobin DNA. But if you substitute that T for an A in that sequence, well, it gets coded by the mRNA, the messenger RNA, a little bit differently, and it causes, instead of a glutamine, a valine being added into that amino acid sequence. And because you've added this different amino acid, it can change the phenotype of the molecule, producing the sickle cell version of hemoglobin, which has medical implications. So an example where just a substitution of a single base can have a big effect on phenotype. Most of the time, no effect at all. So a mutation is defined as something that occurs in less than 1% of a population. It tends to occur in individuals more so than in subsets. But if a mutation or a particular genetic variant becomes more prominent and exceeds that 1%, it's often called a polymorphism, right? And there's a lot of examples of what we call SNPs, or single nucleotide polymorphisms. And the best way to think of polymorphism is it's a mutation that hangs around the population and hangs around from generation to generation. So another way to think of it is it's fixed in the population, right? So you'll have situations where a population may have multiple versions of a protein, and each of those would be a different version would be a polymorphic protein. So another thing that could happen is a big change at the gene level, right? So you can have mutations that occur that change the nature of an entire gene on a chromosome. You could remove genes that are bad. You can make a new gene. You can duplicate a gene. You can potentially recombinate a gene by adding specific segments or deleting specific segments of DNA. And these will have bigger impacts on the phenotype, right? So here are some examples of deletion, duplication, inversion, as well as translocation, where two chromosomes undergo crossing over, and there's basically a switching of the genes that are present on each chromosome. So this is something that can have a very big impact on phenotype, and often when this occurs, this could be the initial stages of formation of a new species, for example. So changing the gene number or the position is another example of the introduction of genetic variation. And that's what you see here. We've talked already about antibiotic resistance. This is a famous example where they visualized a large agar plate, and they basically, in the middle, in the black area, they introduced an antibiotic. And what you're seeing with the arrow and that sudden spreading is where the bacteria first uh, mutate into the antibiotic resistant form. And as soon as they mutate, all of a sudden they've overcome the antibiotic and they're undergoing very rapid reproduction as a result, right? So they're undergoing very rapid evolution and they're basically producing an antibiotic resistant bacteria. Another way to introduce genetic variation is simply by reproducing sexually. Sexual reproduction, when it evolved, which it evolved multiple times, but when it first evolved, it was in many ways a very significant impact on the history of life. It was coincided with all of a sudden this pulse of biodiversity. And the reason is because all of a sudden the ability to produce new variants essentially increased by many fold, right? Now, we don't know much about the evolution of sexual reproduction. Very likely there was, it probably initiated in some way through sort of DNA repair mechanisms inside of a cell. And this kind of stuff got co-opted since they were already interacting with DNA and able to replicate and fix things. And this was probably the beginning of DNA replication for the purposes of sexual reproduction, right? So a lot of sources of variation in sexual reproduction, chromosomes alignments and meiosis, mating, selection, not random mating, sexual selection, right? But the source of the genetic variation is going to be the same. In the end of the day, it's going to be those mutations. But it's a new chance to recombine things in new and unique ways that can potentially confer a huge advantage. But there are situations where sex does not necessarily produce the winning combination, right? So, for example, Deloid rotifers, these are small organisms that undergo parthenogenesis, basically asexual reproduction. You basically clone yourself in a way, and you have the development of a diploid organism that undergoes sort of recombination with its own genotype. And the end result is you produce a fully diploid offspring that is a clone, but can also potentially introduce some new genetic variation through recombination of chromosomes in a particular way. Parthenogenesis is a very interesting sort of exception, but it is pretty widely spread, at least in the animal kingdom, because you have many different groups of animals that are not necessarily related to each other that can potentially engage in parthenogenesis, even if they normally would do sexual reproduction. So many shark species, lizards, snakes, birds, a lot of examples where animals are kept in captivity. There's absolutely no contact with any other animals of their own species. Normally they would reproduce sexually, but all of a sudden they get pregnant and produce offspring. So they're producing offspring because they can essentially fuse their gametes and produce a diploid organism that way. Interesting when it happens. So again, here's our agents of evolutionary change or forces of evolution. Now we're going to talk about genetic drift. Genetic drift is exactly like this picture suggests. It's basically you have a population that's big and it finds a new area, and because of a simple random sampling, you end up with a decrease in genetic variation. So it's really a statistical thing more than anything else, right? So in genetic drift, you're changing the allele frequency of a population, but it's because of sampling, and the sampling being random. So a large population suddenly becomes smaller. It could become smaller in one of two ways. There could have been some kind of disaster, and you end up with survivors, what we call the bottleneck effect. An example of this is what happened to a lot of species where you see decreases in genetic variation in their populations that survive human overhunting, like this cheetah here, right? There's an example of a king cheetah, one of the larger cheetahs found in southern Africa. Cheetah numbers, like many predators, have declined over time because of overhunting, and as a result, you end up with small populations of survivors, and they've lost so much genetic material as a result. You could also have a situation that's less drastic, less disaster based, where you have a small population that settles in a new area, starts to colonize a new habitat or a new island or peninsula or whatever. And because of the few individuals don't necessarily have all the genetic variation of the parent population, then those that end up finding this new colony, if you will only have a small sample. Again, a sampling thing. So is genetic drift, does it affect more a larger population or a smaller population? Well, it depends on how you look at it. If the parent population is a large one, then genetic drift would likely have a smaller subset of the genetic variation that's available, right? But if it's a smaller population and you're sampling, then you likely will get a little bit more proportional amounts of genetic variation. So really, it depends on how you look at it. So it depends on the size of the parent population, and it depends on the size of the survivors or the founders. And basically, it's determined by the proportion of which they've captured a representative sample of the genetic variation of the parent population. So here's an example of a drastic bottleneck effect. This is the greater prairie chicken. The greater prairie chicken. Many years ago, I worked in Minnesota and North Dakota, and we were doing grassland bird research. And I was bragging to the people that I was working with about how I had never hit an animal with my car. And as karma would have it, I later that afternoon hit an animal with my car. And lo and behold, it was the endangered greater prairie chicken that I hit. And it's very unfortunate. It's very unfortunate. I felt quite bad about it because there's not too many of them left. So the greater prairie chicken used to inhabit a lot of the tall grass prairies of the United States. So a lot of sort of the easternmost thing that we would call prairies. Obviously, tall grass meaning the grass is particularly tall. And like many things in the grassland, as it got converted into agricultural farms, you started losing a lot of the native species and their populations got very well reduced. So this is an example of a major bottleneck. So you see here in the state of Illinois, the range of the prairie chicken previously, and then the range later on in the 90s. They have slowly come back. There have been a lot of rewilding efforts, which have been working quite well. Um, but you could imagine that all that genetic variation was lost, and now you're stuck with this tiny population. So that's an example from the animal world of something like this, right? You have an original population. All those marbles in there, the yellow, the blue, and the white represent genetic variation. If you were to make the marbles go through a literal bottleneck and you just sampled a few of them and those are your survivors and all the rest died, you could potentially lose some of the rarer ones, right? Like the yellow variants, you've lost those completely. So your surviving population is going to start and spread and expand, but without any of those yellow marbles, right? So that's an example, practical example of what it would look like. Sometimes documenting genetic drift in nature can be tricky, especially if sampling removes a particular trait. The same sort of thing can happen with selection. So for here, for example, you have a population up at the top and you have some brown frogs. And let's just say the brown frogs, by some calamity, were removed in the population. And the survivors survived and they had offspring. You've lost that brown phenotype. If you were studying this population, you might, if you didn't know about the genetic drift event or the bottleneck effect, you could be forgiven for thinking that maybe selection is acting very harshly on those brown frogs, even though they were removed randomly. So it can be a tricky thing to document in nature. But again, that's an example of a bottleneck that involves death and calamity. You could also get the same exact effect through founding a new population, right? So here's some ladybugs on the mainland there. There's yellow ones and there's red ones, but only a couple of red ones made it to some island off in the ocean there. And the red ones survived and reproduced and had red ones. And now you have a population with a lot less variation than the mainland. So an example of founder effect. And you could also get founder effects in humans as well. So humans, of course, move around, and as they move around, they take genetic variation with them. So we're not exempt from this either. The Ashkenazi Jews are the Jews that occupied Central Europe for a very long time. We'll get into we all know what happened to them. But they have several mutations that are quite unique. So the BRCA1, BRCA2 mutations, these are breast cancer oncogenes. And these are specific mutations that occur at relatively low prevalence in the typical population, but in populations around the world in the diaspora. So this is where Jews move to, Israel, New York, other places from Central Europe following World War II. They brought these mutations, and they actually have a higher incidence of these mutations in their genome as compared to the populations they came from. So again, an example of founder effect, right? All right, so now let's talk about gene flow. So here's a bee moving from one flower to another. It's there to symbolize the idea of immigration from one place to another. And of course, immigration can occur in response to a number of different things. Many factors can influence gene flow. Um, there's the motivation behind moving in the first place, right? So are there resources that are driving it? Are there mating opportunities that are driving it, which can be an example of a resource as well? A lot of factors. The other factors that can influence gene flow are barriers. Are there things in the way? Are there things that prevent immigration and emigration from happening? Gene flow is, at its core, the movement of genes or alleles for those genes from one population to another. In some cases, that can mean depleting genes. In another case, that can mean bringing in new genes, right? So the effect it has on genetic variation can be either, depending. Generally, we tend to think of it here as being something that's positive that brings in new genetic information from somewhere else, right? There's flow. She's all about the insurance. This could be a good insurance policy against losing genetic variation if you're a small population. If you're a small population, as long as you have immigration coming in with new genes, it could potentially keep your genetic variation kind of high, allowing you to deal with the environment as it changes. Right? So it's an insurance policy, and you don't even need to sign up with Progressive to have it. So again, what are some of these barriers? Well, a population is usually defined as being something that is isolated reproductively. So you're a member of the same population if you're, from a biological sense, if you can potentially and reasonably interbreed with other members of that population. But if there is a barrier in the way, that can prevent it, and you'll end up with reproductive isolation. And that's the first step towards speciation, which we'll talk about in a future episode. So this barrier can be a physical one, so oceans, mountains, lakes, walls, whatever, right? So here's an example of a jaguar. There's a couple of jaguars, last I checked, that are roaming around the southwest United States, coming up from Mexico, where they have more of a foothold. And, there's a lot of concern about putting up a border wall along the U.S.-Mexico border and preventing the movement of not only jaguars themselves, but also their prey, right? So it could lead to potentially reproductive isolation on either side of that barrier. In case of humans, there could be other things isolating, societal isolation based on region or cultural, people from the same religion tending to, get married, people from the same class tending to get married, people of the same race tending to get married. So all these different divisions that we have for ourselves, that we've created for ourselves, we can often stay with our own, so to speak, and that could lead to a reproductive isolation as well, right? As much as I'd like to, it is very unlikely that I am going to marry and have kids with some billionaire lady somewhere, right? So I got to stick to my own class. Let's put it that way. Other examples of gene flow can sometimes be muddled, if you will, by the linkage of genes. So if genes are linked, that means they're on the same chromosome, and they tend to be inherited together, right? So there's some kind of as they get inherited from generation to generation, they tend to stick together. So this could help, transfer genes, as opposed to genes that aren't linked often don't exhibit that because they're on different chromosomes or they're on different segments of the chromosome, and they kind of sort independently. I'll also mention horizontal gene transfer, and this is basically when Viruses can move genetic information from, an individual to another individual, one species to another species, or one crop to another crop is the thing about GMOs. And this could potentially, you know, um, also represent sort of a gene flow, an introduction of new genetic material. This is one concern with GMO crops. GMO crops are built to be resistant to pesticides and things like that. But there's always the risk that some of those pesticides resistance genes might make it out of a crop and potentially into another species, a native species, and that can actually have impacts on the ecology of the area, right? If you start being a plant that normally would be pollinated, but you're able to, you know, kill off things because you have some sort of toxin embedded in you, right? A lot of concerns. It's actually a legit concern regarding GMO crops, how they're used, and how biotech is used in general in an ecological setting. So let's talk about natural selection. This is the one everybody knows, everybody understands, survival of the fittest and all that. So when we talk about fitness, we're not talking about being able to do 100 burpees in the drop of a hat or being able to run a 5-minute mile. That's not the kind of stuff we're talking about. Fitness at its core is defined as the reproductive success of an individual and that individual, thus its genotype, right? Because that's the part that's inherited for the most part. So, fitness is kind of this general term to describe a whole bunch of things, right? Your genetic makeup, the phenotype it produces, as well as other impacts of your environment. So that's your fitness, right? So this is showing kind of how phenotype is related to fitness. Fitness is impacted by the environment, right? What you could be fit in one environment and not very fit in another environment. And environment is also going to impact the phenotype through impacts on development, through, even if you have genes and genetic effects, they can be altered by the environment. This is where we get in the realm of things like epigenetics, for example, right? All this stuff is it's kind of a construct, right? But in the end of the day, fitness is going to be the thing that natural selection is going to work on, right? So your fitness is what natural selection is to hone in on. And this could take different ways, right? If you look at the range and variation of a trait in a population, if selection is occurring, you can basically see evolution over time. And if you keep sampling over time in a consistent sort of way, you can start to pick up on the different types of selection. So here, for example, you have variation in fur color in mice. So in the first example, you have an original population that's showing that typical bell curve. So most of your individuals are going to have that middle phenotype, right? They're not going to be too light in color, they're not going to be too dark in color. But if selection is acting on that original population, which is shown here in the dashed line, and let's say there's strong selection against having a light color. Maybe you stick out in the environment and you're more likely to get caught. So what's going to happen? Well, the entire population, the average, and the variation, the standard deviation, for example, that whole bell curve is going to shift to the right. It's going to shift with the average now becoming a little more darker than the original population. So that's your new evolved population. Over time, you're going to see this as being there's more dark mice in your environment. So that's an example of directional selection. The effect on genetic diversity, well, it's going to decrease over time because you're losing those light-colored animals, right? So you're shifting it, but at the same time, it's still a bell curve. So you might end up with new mutants that are even darker than the furthest end of your spectrum, right? But over time, this can lead to potentially to variation in a species, right? If it's going to lead to a new species, well, if enough time goes by, possibly. So a second type of selection is disruptive selection. Again, you have an original population, standard bell curve, but now you're selecting against the middle of the road, the average, the most common types of individuals in your population. So it pays to be on the extremes now, right? It pays to be a light-colored mouse, it pays to be a dark-colored mouse, but you don't want to be something in between, because if you're something in between, selection is going to act on you. Over time, that's going to essentially decrease your genetic diversity, but It's also going to, or it's actually, sorry, it's actually going to increase your genetic diversity because you kind of spread out and you're starting to get more variants. So what was previously rare variants of your mouse, your white and your dark mouse, are now going to become more common in your population. Now, as disruptive selection keeps working, you're disrupting that bell curve and you're going to produce eventually, long enough if this happens, you have an increased chance of producing a new species, right? Simply because you're going to have mice that use their light color to one advantage. You'll have mice that use the dark color to another advantage. On the flip side of that is stabilizing selection. Stabilizing selection is what happens when you have your most common phenotype being the most selected and your extremes are being selected against. Over time, that's going to make your bell curve even narrower. You're going to decrease your genetic diversity and you're eventually going to start losing that variance on one side or the other, right? New species, very unlikely. All you're doing is kind of reinforcing that most common phenotype. Now, sometimes selection is going to work depending on the current condition. So here is a fish, African scale-eating fish. It has a mouth that's kind of oriented in a way that it's able to latch on and take bites of fish while they're swimming around. So it actually attacks its prey. And based on the side that its mouth occurs on, it's going to bite prey on that side. So you have a left mouth phenotype and you have a right mouth phenotype, right? And they're both inherited roughly equally, meaning that about 50% of the population is going to have either one or the other. But here's the thing: it pays to be the it pays to be the one that tends to be rarer, because the fish aren't stupid, right? If the fish is constantly being attacked by the more common one, say on the right, that fish is going to anticipate being attacked on the right, and this is going to make it harder for that one to use its element of surprise to take a bite out of it. As a result, it's better able to foil the plans of the right mouth fish, but the left mouth fish, well, has an advantage now because it's not anticipating an attack from that side. So what's going to happen is as time moves on, the left mouth fish is going to increase in numbers in the population. But the animal, the prey, is going to learn. And it's going to now do the same thing for the left mouth fish. And thus being better able to stop it. So now the right mouth fish has an advantage. And this goes back and forth, back and forth. So what ends up happening is that in any given year, you're going to hover up or down around 50% frequency of individuals for these different phenotypes. And whatever one happens to be rarer will be the one that at that time has selection going for it. So what that means is it's frequency dependent. The frequency with which a phenotype is present is going to determine how selection is acting on it. So frequency dependent selection. So a thing to remember is it's very tempting to look at something like a hummingbird on a flower and it's pollinating it, to see it as this perfect combination, a perfect adaptation. No organism is perfect. There is always something that selection is acting on. Selection is only going to work on the variation that is present, right? So what ends up happening is over time, evolution is just going to hone the variation that's already there. Nothing new gets made except by mutation, and even then it's a very slow process before you produce something that you can see. So we tend to think of these perfect adaptations, but the reality is that there's usually a compromise, and selection can change over time depending on the environment. So things that we see as adaptations are often come with costs. So an example would be having flexible limbs and the prehensile hands and their ability to carry things. Well, that's great, but that's also why we're uniquely susceptible to things like ligaments being torn, sprains, dislocations. This is all a product of having adaptation honed for a particular purpose, us using our hands for something, right? Other animals don't have those issues, right? Because selection is acting on them differently. So everything has you know, costs. In the case of hummingbirds, they rely on a very high sugar diet. They have an extraordinarily high metabolism, but that also leads them very susceptible to things like heart attacks. It leads them very susceptible to things like starvation. They have to feed all the time, or else they have to somehow enter into a torpor state to be able to survive even one single night, right? So there's also big selection on their migration. When they do migrate, they have to make sure that they have their food sources available because they'll burn through those reserves very quickly because they have such a high metabolic rate. So there's nothing such thing as a perfect organism. So let's talk about non-random mating. And here's the classic picture in all of your textbooks of a male peacock trying to impress a peahen with his long, large, beautiful, colorful, elaborate tail. It's doing this display, and conventional wisdom will tell you that the bird with the best display, the best feathers, the best everything is in better condition and thus have a better chance of mating. So the mating is not random, right? Unlike, say, drunken college kids, mating is going to be determined by factors, right? A female's investment in offspring is quite substantial, and so she wants to make sure that the best traits are available to her, right? And this is a fundamental asymmetry between the sexes, or between the male and female genders. The biggest issue for females is they have to invest so much in eggs and in young and in parental care in some cases. So they want to make sure they gain the resources to optimize the likelihood that those offspring will survive. In males, the fitness issue comes from the fact that they have to compete for mates. They either have to compete directly between themselves, right? So intrasexual selection or male mating competition. So here, for example, you have two ibex that are basically banging heads, trying to gain access to a harem, right? So these are the ancestors of the modern goat, and they're battling it out. And the tougher, stronger, better fighter is going to be the one that gains access to the harem of females and thus produces the offspring. On the flip side, you have the female making the decision, the female choice, intersexual selection. Here, males will compete for females' graces or females' interest, often through elaborate courtship displays. So here's a mannequin, for example, under doing this little dance to a female. The female is obviously the drab one because she's having to take care of offspring and needs to be able to hide. The male, on the other hand, is very showy. In both cases, the success of both the ibex as well as the mannequin is going to be determined by their condition, right? Which one's in better shape, right? Which one can dance the longest, which one can fight the best. So nature tries its best to preserve genetic diversity in a variety of different ways. And there's always mechanisms to keep things going, right? Generation after generation. First, if you're diploid, right away, if you're diploid, well, that means that you're going to hold on to recessive alleles in your population by hiding them in your heterozygotes. So, for example, here's the classic example. Here's malaria, resistance conferred by having sickle cell disease, right? So sickle cell causes your erythrocytes or your red blood cells to take on a little moon-like shape, right? And as a result, it impairs your ability to transport oxygen, impairs your ability to develop hemoglobin, and it impairs your ability to combat. It just generally makes you sickly and unhealthy, right? However, the fatal disease is something that occurs only in the homozygous recessive condition. So in a homozygous recessive condition, all of your red blood cells are like that. So this is essentially a 100% fatal disease, barring some treatments now that are available. Now, if selection was acting on this, well, if every time you get the sickle cell, it should die off very quickly, right? If it's fatal, then it should be eliminated from your population over time. And yet, sickle cell is still an issue simply because we're dealing with diploid organisms. And in diploid organisms, you always have heterozygotes. In the case of heterozygotes, half of they're an example of codominant. So half of the red blood cells are going to be the normal sort of donut shape, and the other half are going to be this sickle cell shape. Well, it turns out that the heterozygotes, not only do they keep those genes for that sickle cell constantly in your population by hiding them in these heterozygotes, and then later on they'll resurface with mating. But it also confers a bit of an advantage. So you get resistant to malaria because malarial mosquitoes and Anopheles mosquitoes will prefer not to feed on blood cells that are like this, right? So they actually prefer the homozygous dominant condition of quote unquote normal blood cells, right? So as a result, you get a certain resistance to malaria from that. Now, another example where nature is preserving genetic diversity is by having balanced polymorphism. So we already mentioned the heterozygote advantage, right? So that's an example of the heterozygotes being advantageous in this case. Frequency dependent selection is another example where you have multiple versions of a trait, and because you have multiple versions of a trait, well, depending on the current conditions, it'll survive, but as it becomes more common, it gets selected against and back and forth. Nothing ever gets completely removed from your population. The third way nature preserves genetic diversity, we haven't really talked about it, but most of your genome is not doing anything that we know of particularly, meaning that it's not necessarily coding for a gene, or it's not necessarily a gene coding for an allele, coding for a protein that's going to produce something. A lot of it is involved in regulation of gene expression, for example, but a lot of it's also, up until now anyway, we will call it a non-coding area or non-coding region. So these are areas where there is genetic diversity, but they're not under any kind of selection because they don't produce a product necessarily, right? Over time, these could become something more important to the organism, but it does provide another way to preserve genetic diversity. And a lot of these non-coding regions are often used as markers of a population strictly because they're not under selection and they're not producing anything. So they allow you to sort of blindly trace things through time without, as a control, without having to be affected by anything in the organism and what it's doing. So genetic diversity matters. This is the seed vault located in Norway in the island of Svalbard. And it's the seed vault because it's where we store the world's diversity of seeds. So there's entries made, it's deep underground. I think it's like 4 floors underground, deep in the ice there. And the idea is that crops all around the world tend to be monocultures, meaning that they have a very low amount of genetic diversity. They're often derived from just a few founder plants. So it's an example of genetic drift. And because of this low genetic diversity, they're very susceptible to environmental changes, right? And of course, with climate change on the horizon, we often call this the doomsday vault because, every crop has different breeds, different versions, different culture types, and all of those are found in the doomsday vault. So the idea is by preserving the world's seed diversity, we could potentially have the variation that's necessary to cope with environmental changes as a result of climate change as they come, right? So even though you may never have thought of genetic diversity before this, I assure you that other people have, and it plays a very important part in our biology and a very, very important part in evolution. Until next time, have a good one.